Very good morning, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. So, we would like to invite today's eminent resource person, Dr.A.Rajani Madam Garu, Assistant Professor, Head Department of Botany, Food and Nutrition, Rbvrr Women's College, Autonomous, Hyderabad. Madam is going to explain today one of the important topic in algae in Chlorophyceae member, that is Chara. So, Madam, please welcome. Start your session, madam. Welcome.
Okay, sir. Thank you. Are you visible, my slides?
One second, madam. Already I have given sharing option. Okay, I have given to you.
Okay, okay, sir.
Screen sharing I have given. Your slides is clearly visible, madam.
Okay,
Please
okay,
click
sir.
on slideshow. Please click on slideshow.
Yeah, yes.
Yeah, madam. Okay, it's clearly visible. Please proceed.
Yes. Good morning to one and all. Today I'll discuss about the topic name is called the Chara. Here in the last class already to study the Chlorophyceae members, 2 type studies are completed. The first type study is called the Volvox. The second type study name is called the Idogonium. Both are completed. Now the third type study in same class, Chlorophyceae member, that is the Chara. Here, what is the Chara? What is before Chara explanation? In the Chlorophyceae member, already you have studied to the 2 type studies. What are they? The first type study is called the Volvox. In that Volvox, the plant structure, what is the thallus is there? In the thallus, very minute thallus is present. That is a cenobium colony is there. You can observe only under electron microscope only. This is the Volvox. Cenobium colonies are present. The thallus structure you can't see directly. Only you can observe to the under electron microscope. This is a first type study, the Chlorophyceae member. The second one is the Idogonium. The Idogonium, what is the thallus structure? Here, filamentous body is present, unbranched. To compare the thallus, both the Volvox and Idogonium. Volvox fully colony type, cenobium type. Here, the plant structure or thallus is called the filamentous plant body and unbranched. Branches is totally absent. 2 parts most important, holdfast and cap cells also. These 2 are main important character, the thallus structure. The third one is Chara. Why I'll to both to again to explain. The Chara thallus is very fully different. This is the macroscopic structure. Chara is the macroscopic structure. You can see directly eye contact. You in Volvox and Idogonia, you can't see in directly. You can observe to the under electron microscope only. But Chara, this is a macroscopic structure. This macroscopic, see here, this one is the Chara. No, Chara diagram. See, this is the Chara picture. You can observe the very directly eye contact. That's where in this Chlorophyceae member, very macroscopic algae can ask the question, which one is the macroscopic algae? That is called the Chara. See, here Carl's order sometimes to classification also to ask the question. That's why you can byhead it each and every classification also. Same Chlorophyceae member, order is Carl's, the genus family Characeae, the genus name is called Chara. Okay. Here, before explanation of Chara, here some introduction of this one. Some important points are there. In Chara, here another names also there. Stonewort, oblique, brittlewort. The Chara, these questions to expect questions. Which algae is the stoneworts? Which algae is called the thallus is the brittlewort? Why this stonewort and brittlewort? See, Chara stone what grows. This is the Chlorophyceae member, no? That's it. This is a fresh water, clear, and standing waters on the muddy or sandy bottom, sometimes below the water level. It can also be collected from standing or tangent reservoirs also. A few species grow in brackish water. That is a Chara balitica. This is a brackish water, meaning just like saline water, salt water. Only Chara balicta grow on salt water. Coming to the vegetative and what is the thallus structure? The common name of Chara is called stoneworts and brittleworts are suggested because they are encrusted with calcium carbonate. The plant body made up of to the calcium carbonates. That's where the name of Chara is called stoneworts or brittleworts. You can touch the Chara thallus little bit very rough surface is there. Why? The total thallus is made up of the calcium carbonate. That's where the name of the Chara is stoneworts and brittleworts. Keep in your mind, very, very important. The ask the which algae is the thallus, is the stoneworts or brittleworts or made up of the calcium carbonate. This one. You know all all algae, the cell wall made up of which one? Cellulose. But here calcium cell wall meaning the total cell wall to made up of the big thallus. No, see converted in thallus. But here the calcium carbonate is very dominant. That's why you can touch the Chara, brittle words and stone words, very rough surface is present. See here, this is a Chara examples. Chara vulgaris, Chara jailanica, Chara nuda. These are the examples for Chara. See, you can await the occurrence. I'll already explanation is complete. No, Chara baticula grow on brackish water. Chara fragilis is found in hot water springs. See, these two you can buy had it. All are the found in the freshwater, lakes, tanks, reservoir like this. Only two species to found in salt water or brackish water, Blatica, and hot water streams or hot water springs to found in Chara fragilis. This Chara mostly hot fresh water, rich in organic matter, calcium and deficient in oxygen. The Chara plants encrusted to made up of calcium carbonate. That's where this commonly called as stoneworts. And another one here, Chara emits disagreeable onion-like odors. The smell of that one presence. Why? Here the sulfur content is more, compounds are present. That's where this is the odor. Onion-like odor is present. In India, Chara represented to identify the scientists 30 species. But best example, Jailanica, Baruni, Gracilis, and Hatei. These are the present in our India in the Chara different species. And Chara, what is the thallus structure? The thallus is macroscopic, branched, and multicellular. You can observe the picture. Ma. That one is, see this point. This is the Chara thallus. This Chara thallus is the macroscopic. You can see directly, no? Macroscopic, branched, and multicellular. The thallus is very most important, erect, branch, axis attached to the substratum by rhizoids. Substratum by the rhizoid structures are present. See, you can observe, this is the rhizoid. What is the rhizoid? See, this is the Chara total plant. 1 second. See, this is the structure, no? This plant, the thallus is the branching are there, okay? Multicellular branching. Main axis also present. In the bottom, you can observe The substratum is present by rhizoids. See, this one is the length of algae, the length of Chara is a minimum 20 to 30 centimeters length. Main axis is present. In the bottom, you can observe rhizoids. Main axis, each main axis, here branches are there, and some, this one is a branches. Branch and main axis contain the nodes and internodes. Node is a 2 central cell, 6 to 20 cortical cells are present. The another one is a substratum is attached to the rhizoid. The rhizoids are always whitish color, multicellular, branched, borne on the lower node part. See how to born this one? Lower node part of the main axis to produce the rhizoids. The rhizoids possess oblique septa. This rhizoids is a possess oblique septa without differentiated into nodes and internodes. Here, the node differentiated in the node, we can only observe the way the axis part is there. In that one, you can observe the node part and internode part, but absent in the rhizoid side. Where the erect axis, this is a, what is the main characteristic of the rhizoids? Help in attachment of the plant any substratum, that is called muds or sand or absorption of minerals also. And vegetative time, vegetative reproductive time, multiplication of so plants by forming bulbils and secondary protonema. This is the function of the rhizoids. I think you can understood for the rhizoids. See, what is the main axis? This is the main axis of the Chara. The erect axis as equisetum-like differentiation. You can observe the in the equisetum, plant of the equisetum. That one is same Chara plant and equisetum plant. Some plants is a similar. The erect axis, you can similar equisetum-like differentiated into small node and long internode. Where the node part, this is the node part. Where my arrow is there, that one is the node part. This one, this long internode, this part is called the internode. That's where small nodes and long internodes are present. That is a nodes and internodes are both are present. And a node is a group of irregular arranged cell is called the node part. And form the nodal region, two types of branches to produce the node part. What is that from two types of branch? One is the, see each and every node. See in this one is the main axis. This part is called the node part. In between node, this part is called the internode part. Where the node part is there, in picture you can observe. Where the node part is present, the node part region, two types of produced branches. One branch, lateral branch of limited growth. Lateral branch of limited. This is the lateral branch of limited growth. And second one, lateral branches of unlimited growth. See, this one, where the lateral branch is there, that is the limited growth. Second one, lateral branches of this part. One is the limit growth. Second one is the unlimited growth is there. Limited growth are in rows around nodes of the main axis. See, here main axis, erect, long, differentiation into the node and internode. Internode consists a single, much elongated or oblong cells are present. Where the internode cells are there, no? Some species surrounded by the one cell thick layer cortex is present, that is the corticate. Some species cortical layer is absent, that one is the acorticate. Okay, we need internode part. Some species cortical cortex layer is present. When the when the cortex region is present, that is the corticate. Where the cortex is absent, that part is called acorticate. The node, you can observe central small cell surrounded by 6 to 20 peripheral cells are present. The central cells peripheral arise from the single node. This is the main axis. Coming to the node part, to produce the two type branches. No, one is the branches of limited growth. Another, this one is the branches of, one is a limited growth. The second one, branches of unlimited growth. Branches of limited growth, you can observe this one. The whole 6 to 20 from peripheral cells of the node of the main axis are branches of unlimited growth. This one. These are the called bractlets. Branches of first order is called the primary laterals. This one is these are the primary laterals or leaves. Just like this one is called the primary laterals or leaves are present. These branches, you can observe not this one. You can observe the where my arrow mark is. This is called the primary laterals. The branches of limited growth. These are the branches of limited growth. That is called the primary laterals or leaves. How many to produce? 6 to 20 minimum. These branches stop to grow after forming a 5 to 15 nodes, and hence it is called branches of limited growth. Here you can observe the stipules and reproductive structures are formed the node of these branches also. Coming to the branches of unlimited growth. See, this total is unlimited. This one also unlimited growth. This one also unlimited growth. The branches of unlimited growth arise at the axil of branches limited growth. Hence it is called axillary branches or long lateral branches. These lateral of unlimited growth arise from the nodes of the main axis only. These are situated in the axis of short laterals. These laterals of unlimited growth arise, growth process a same characteristic as that of the main axis. And these are differentiated also node part, and this unlimited growth also same like that main axis. Nodes are there, internodes where the node part is there, and bears primary laterals. And these branches look like the main axis only. Their growth is also unlimited growth like main axis. See, this is the main axis. What is the stipulodes? See, you can observe this is the main axis. This is one is the main axis. You can observe the external morphology, this total. In this one, this main axis, this is the short of unlimited growth, branches of unlimited growth. In this one, in the unlimited growth, these are the primary laterals. This is the limited growth. This branch is the unlimited growth is present. Okay, here many stipulodes also present. What are the stipulodes? Here, the basal node of branches of limited growth develop very short ovule pointed like structures are present. That one is called the stipulode. See, you can observe this one. You can observe to the appendages of nodes. This is a node part, no? In that node part, some pointed single cell oval shape outgrowths are present. These are called stipulodes. In most of species of Chara burmanica, number of stipulodes at each node twice the number of primary laterals. Listen careful. Burmanica, the number of stipulodes at each node is twice the number of the primary laterals. Such type of species name is called bistipulate. In some species, Chara nuda, Chara burrini, number of stipulodes at each node is equal number of primary lateral. That node, such species are called unistipulate, bistipulate, unistipulate. Some distipulates also present. When the stipules are present in one hole, the each node part is called the haplostiphonos, and which two rows of the each node is called diplastostiphonodes are present. This is the total structure of the stiphonodes. And what is the cortex? Many species Chara, aspera, Chara infera, internode cells of main axis, end-sheeted and cortical cells are present. Such type of species are called corticated species. The cortex consists of vertical and elongated narrow cells are present, where the internode part of the internode corticated filaments develop from the upper node. This is called descending layer of the internodes. This is converted into the filamentous developed from the lower surface of the filament. This ascending and descending filaments, just like the T shape are there. This one is the T shape. These T shape example Corallina cara are called E-corticated species are also present in the Chara. See, the cell structure very thick. Three layer, inner layer made up of cellulose, outer layer made up of pectins and calcium carbonates. Two layers of cell wall is present. Outer cellulose, inner cellulose, outer pectins and calcium carbonates. That's where this is what is the brittle little bricks are, that is the name is called stone words and brittle words. And haploid structure is there. The chloroplast is the discoid type of chloroplast is present. The cell possesses numerous discoid chloroplast without pyrenide. Very, very important. Chloroplast is there, but pyrenides are absent. Sometimes to ask this question also. In which chlorophyceae member, which algae pyrenides is absent? In Chara, chloroplast is there, but which one is absent? Pyrenides are absent. What type of chloroplast is there? Discoid chloroplast are scattered throughout the length of cell or in the peripheral layer of the cytoplasm. A single nucleus present at the center of the cell. And coming to the, what is the node cell? What is the internodal cells? Node cells small in size, isodiametric, very rich in cytoplasm. Single nucleate is there. Small ellipsoidal chloroplast are present. Central vacuole is not developed. Instead of many vacuoles may be present. Cytoplasm can be differentiated into outer and inner. This is a nodal cell structure. Internodal cell structure, cell much elongated. This is very small, no? This is fully opposite, very much elongated. Cytoplasm also present around the vacuoles also present. Multinucleate. Contain the, what type of chloroplast here? Discoid chloroplast is present. Here, ellipsoidal chloroplast, nodal part. But internodal, discoid chloroplast is present. Cytoplasm same, differentiated outer and inner. Endoplasm also present in this one. Coming to the reproductive structure. I think you can understood the internal morphology, internal and external morphology. Yes? Any doubts in that part? No doubts? It's clear? Message me, ma. Coming to the next one, reproduction part. What is the reproduction here? Main vegetative reproduction is the most important in this one to expect the questions here also. Okay. What is the, many times to ask this question also. Where the amylum starts vegetative reproduction present in algae. That is the Chara. Here, more 5 types of vegetative amylum starch or amylum stars. Example, Chara stillageria. Chara stillageria is the present in the vegetative reproduction. Bulbils, second one. Amorphous bulbils, third one. Tubers, secondary protonema. See first bulbils, spherical shape or oval shape, tube-like structure. Which one is to develop? Rhizoids to develop bulbils. These bulbils detach with the plant, germinate new Chara plant. Example, Chara aspera. Coming to the amylum starch. See, this is a very, very most important amylum starch. Keep in your mind or write down this point. The vegetative reproduction amylum starch you can observe in Chara, only Chara staginella. Some species of lower node part of main axis develop multicellular star-shaped aggregations of cells are present. These cells filled with the amylum starch. Hence, it is called the amylum stars. The amylum stars do the attachment from the plant, develop into a new Chara plant. This is the amylum starch. Very important. Keep in your mind this amylum starch. Third one, amorphous bulbils. These amorphous bulbils also many cells irregular in shape, lower node main axis to produce these amorphous bulbils. Example, Chara delicuta or Chara fragifera, Chara blatica. These are the example for the amorphous bulbils. Penetrating structure, this one, main plant die under unfavorable condition. In case unfavorable condition is there, in that time main axis is die. After favorable condition, these bulbils survive and make Chara plant and return on favorable conditions. And next very important, another one is amorphous also very important. Coming to the next one, secondary protonema. See, this one is also expect this one. Secondary protonema. These are tubular filamentous structures developed from primary protonema of basal cell of the rhizoids. These also rhizoid basal cell to produce to the secondary protonemas like primary protonema from the Chara plant. See, this is the total vegetative reproduction in Chara. See here another very important point also there. In Chara, asexual reproduction is absent. Only which reproduction is there? Two reproductions. One is the sexual, first one is a vegetative, the second thing is the sexual reproduction. Which one is absent? This is also important. In Chara, which reproduction is absent? Asexual reproduction, spores not produced in this Chara. That's where the asexual reproduction is totally absent. Sexual reproduction type is oogamous type. Majority of species are homothallic, that is called monoecious, while few are heterothallic, that is called the dioecious. Monoecious example, Gylanica Chara. Welwitschii is the dioecious species. Male, another very important point. The sex organs arise on the branches of limited growth or primary laterals, the nucleus and the globule. This question also many times to us. The male reproductive organ, very easy. Antheridia archegonia, not Antheridia archegonia. Here the names are most important. Keep in your minds. That is the male reproductive organ and female reproductive organ names. Male is globule, female is nucleus. Both globule, nucleus are born on one point on the node of the short lateral, which bears stipules. The nucleus is always born above the globule. See, this is the nucleus. This one is the globule. The nucleus is always born above the globule. The globule is a very small spherical body conspicuously red or yellow in color, attached to the node by long stalk cell. Nucleus, nucleus is oval in shape and situated above globule at the node part. See, the sexual reproduction in Chara, oogamous type, sex organs are called, antheridia is called, what is the antheridia name? Globule, oogonium name, nucleus. The male reproductive globule, female reproductive is called the nucleus. At node, globule is present at the lower side of nucleus is present at the upper side. Most of species are homothallic, some are is the heterothallism. First, I'll explain to the what is the globule. That is globule, antheridium, spermatangium is very common. But the name you can by heart, globule name is most important for the name of male reproductive organ. See, the antheridium or globule is very large, hollow, bright yellow color or a bright yellow or sometimes reddish color also present. Here, the wall of antheridium is generally mode of very important. Listen very careful. This point also to ask the expect the questions. Here, the wall of antheridium or wall of globule is generally made up of 8 triangle plate-like cells is called. Triangle, how many cells? 8 triangle plate-like cells are called. That cell's name is called shield cell. See, first 1, 2, 3, 4, 5, 6, 7, 8. In the globule, how many shield cells are present? That is called the 8 shield cells. This is the first important characters. The 8 sealed cells actually form the wall of antheridium. This one is the form of the antheridium. Next, from each sealed cell, can observe, from each sealed cell as handle-like cells are present. Handle-like elongated extends into the antheridial cavity. That cell name is called manubrial cells or manubrium. See, 8 shield cells are present. After 8 shield cell, some elongated, handle elongated like cells. This part, this is the handle elongated cells are present. This handle elongated cells name is called antheridial cat. This is name called manubrium cells. See, that is called the manubrium. The manubrium cells, same 8 shield cell, 8 manubrial cells. The manubrial cells terminate with primary capitular cells. See, after manubrial cells, you can observe these in between manubrial cells, you can observe the primary capitulum. This is the primary capitulum. And this one also primary capitulum. This one also primary capitulum cells. The primary capitulum cell cuts off 6 secondary capitular. See, after lower part, you can observe the primary lower. You can observe the secondary capitula cells also present in this one. Each secondary capitulum cell produce a pair of long unbranched spermatogenous. See, this is the secondary one to produce the spermatogenous cells or spermatiums are present. Filaments, each cell produce single anthrozoid. Here filament consists of 1 to 200 cells are produced. Each cell produces single anthrozoid which is biciliated and spirally coiled like structures are present. This is the globule structure. Keep in your mind here 8 shield cell and 8 manubrial cell and primary capitulum, secondary capitulum, spermatozoids or that is the anthroidal filaments are present in this structure of globule. You can by head it the names. Coming to the nucleus. The mature nucleus is a green in color. See, this is the nucleus, no? Green in color, barrel shaped. What is the shape? Green in color, oval or barrel shaped. It is born on node of primary laterals. The next one, the structure with very short stalk also present. This is the stalk, very small stalk or pedicels also present. The oogonium, this one, oogonium or nucleus consists of a single ovum surrounded by, see here, surrounded by 5 spirally twisted tube-like cells are present. That 5 tubes are called corona cells. See in the picture, you can observe only 3. This picture you can observe 1, 2, 3 and 4, 5. 5 crown-like structures are there, no? Same our corona type. These cells, crown-like cells are present. That's where this name is called corona cells. And the oogonium develop from the nodal cell which lies on the adaxial side of the leaf. Oogonial initial starting by 2 transfer division to produce a row of 3 cells. The lowermost cell act as the stalk cell, this middle cell function as the nodal cell, uppermost cell is developed into oogonium. The stalk cell always act as the pedicel without any further division. The stalk cell without any division form to the pedicel. The nodal pedicel, and next one is the nodal cell divides into a central surrounded by a peripheral cells. These are elongated tubular cells are present and small corona cells also present. The upper oogonial cell produced, where the upper cell is there, in the oogonial cell produced oogonia. This one later developed into the form of egg or ovum. The oogonium is enveloped by tubular cell in the clockwise manner. See, tubular cells are there, no? These are tubuloid. Clockwise manner. Finally, the oogonium develop. 5 called corona act as the apex of the nucleus. This is the formation of the oogonium. How to fertilize in this one? When the oogonium is mature, the 5 tubule cells get separated from each other, forming a narrow slide between them. The anthrozoids are chemotically attract to the ovum. These anthrozoids enter through this slit of the penetrate and gelatinous wall from the oogonium. Many anthrozoids enter into the oogonium, but only one to fertilize the egg to form the diploid zygote. The zygote create a thick wall around the make the oosphere is to form. The oosphere, very thick wall is present. Oosphere is the very thick wall. And that is the inner wall tube cells thickened is present. The germination of the oosphere. See, this is the after germination of oosphere, how to the development of Chara. The oosphere of Chara do not germinate in dark. They require white or red light for germination time. At the end of resting period, the diploid nucleus moves to the anterior end. The oosphere divide meiotically from 4 haploid nuclei. The transfer wall is now laid down near the anterior end, forming to the 2 cells. The upper cell is lenticular uninucleate. Lower cell is large trinucleate. Upper cell divide vertically to form to the rhizoid. This one, rhizoidal initial and a protonemial initial. Both these cells grow in opposite directions. You can observe the opposite directions. The rhizoidal, see, the rhizoidal initials show positive geotrophism and form primary rhizoids, where like protonemial initial shows negative geotropism and from the primary protonema that the primary protonema differentiated, you know, that is the nodes and internodes. Rhizoids is the secondary protonema, arise from the peripheral cells of the basal node. The peripheral cells upper node gives rise to the lateral branches. This is the development of germination of the oosphere, or the what is the plant body life cycle here? Alternation of generation is there. The plant body of Chara is haploid. The diploid condition is found only, you can observe oosphere time. At the time of germination, the diploid nuclei of oosphere divides meiotically and thus haploid states to regained. That's where the Chara life cycle is haploid is predominant type of the position. This is the total study of their Chara type study. If you want to ask any doubts, I'll clarify. Yes? అర్థమైందా అందరికి?
Yes, my dear friends, I have given a chance to unmute yourself, my dear friends. Regarding of this class, if you have any doubt, please interact with our respected madam. Okay, my dear friends. Prakash, Nagireddy, Maria Mahevin, Nafis.
No doubt, sir.
Okay.
No doubt, sir.
It's clear.
Okay. So during this class, madam has clearly explained about the beautiful, wonderful structures. And excellent structures we are observing nucleus and globule. These are the two sexual reproductory organs we are observed in this Chara. And again the anatomical structures of the sexual reproduction organs is most important in this class. And also the protonema, development of the protonema, and also the meiosis, the takes place the zygote. So zygotic meiosis we are observed in this Chara, and also the other names of this Chara, brittlewort, stonewort. Why? Because the calcium carbonate is established in the outer structure of the Chara. It is the macroscopic. Compared to in previous classes, the Idogonium and Volvox, these two Chlorophyceae members are the microscopic, but whereas in Chara, it's a macroscopic. Madam has clearly explained. So, my dear friends, these are some of the important points you note down during the class. So, thank you very much, madam, regarding of this class. I hope all the participants and aspirants, they are very enthusiastically listen, and also they clarify their doubts, and I hope
Okay, sir. Thank you, sir.
the positive participants. Again, 12 o'clock we have another session. Dr. P. Manjula Madam Garu will going to join through same link. So my dear friends, please you join sharp 12 p.m. Okay, madam, thank you.
Hello.
Madam, thank you for this class explanation. No doubts, ma'am.
Yeah, it's okay. Thank you. But the points are most important: brittlewort, stonewort, manubrial cells. And these are the expected questions. Amylum starch. And next one is the after amylum starch, protonemas also sometimes to ask the question. Same reproduction, globule and nucleus also very important. In the globule to ask the manubrial cells. These are the cells are most important. Secondary capitulum, primary capitulum. How many manubrial cells are? How many shield cells are there? In the nucleus also, nucleus, what type of corona cells are there? How many ovumes are there? Okay, these are the important characters. Okay, sir. Thank you, sir.
Okay, my dear friends, my dear Prakash. So cells and primary capitulum, these are the most important in the globule. At the same time, in nucleus, the coronary cells, 5 coronary cells are present. This is the most important.
Okay, sir. Thank you. I can leave, sir.
కషాబాల చేత చెలిస్తాయి గుర్తుపెట్టుకోవాలి మిత్రమా. ఓకే మేడం, థ్యాంక్ యూ.
Thank
టుమారో
you. Yes, sir. Same
Going to
time.
join. Okay, right.